Science is self-correcting. Sometimes we take detours, but we get to the truth, and that's when we find things that maybe can make a difference.
You have to be extremely creative in this line of work. You have to be innovative all the time. This is a very fast-paced field, and if you are not staying up with the new technologies or inventing your own, you could quickly fall off of the map.
Cystic fibrosis was an unsolved mystery at the time I became aware of it. And a lot of our work here has been devoted to trying to crack open that nut. In the last 10 or so years, newer approaches to gene editing have come along, and we're really kind of in a renaissance period of an amazing toolbox that we can apply to the problems.
It's incredibly exciting. I love discovery. I love seeing something that no one's seen before.
I think we're so close, and to see where we've come in 10 years is just astounding to me. There's so many genetic therapies that are going into clinical trials right now. The technology exists, the tools exist, and it's just a matter of time before we find the cure.
When I was in medical school, I decided to go into pediatrics. At that time, there was one investigator in internal medicine at Iowa who was studying cystic fibrosis, and that was Mike Welsh. And so I went and talked with him, and that opened my eyes to what is going on in CF and how can we fix it.
So I'm Mike Welsh. I'm a physician scientist at the University of Iowa. Most of my time right now is focused on research and trying to change the lives of people with CF. CF's an inherited disease, so you have a mutated gene. The gene is the instruction, and we discovered that that is a channel, like a pore, that sits in the cells in the airway, and it allows salt to flow through. That was important because it said there's a roadmap now. If I want to make a drug, I need to know what mutations I'm making it for. And there's increasing emphasis on therapies that aren't mutation specific, they're mutation agnostic. One of those is genetic therapy.
So when I started working in the lab in 2010, we were working on gene addition. A few short years after I started in this field, the concept of gene editing was introduced. The discovery of CRISPR caused the field to explode. And since then, there have been many more advances in many other gene editing technologies. And we use those tools now in the lab every single day. So we have 12 incubators, and they're filled with these trays of lung cells. They're cultured once, and they can grow for years on these filters.
We're testing gene editing tools in cells derived from the lungs of people with cystic fibrosis, and we're seeing very promising ability to repair nonsense mutations or stop codon mutations and restore CFTR function.
So in order to validate gene therapy tools, you have to start with your virus that you design. And so you have to think about what your target is, and if you're doing a gene addition approach, we do molecular cloning. And we'll clone the gene into something called a viral vector, that's sort of like a vehicle or the Trojan horse to get into the lungs. And so then we'll take that viral vector, put it on cells, and then we'll look at the electrophysiology of these cells to see if we've indeed fixed the gene. We can use primers from IDT to make changes in our vector design, and then you can deliver a gene editing tool and fix these mutations.
When we first started working with CRISPR nucleases, we used materials from IDT. Working with Mark and his team, we learned how to quantify gene editing efficiencies using high-throughput sequencing techniques. And we used IDT tools to amplify the regions of the genome that were edited.
So working with IDT, we can reach out to them and say, this is the problem, how would you advise that we can fix it? Say we want to amplify a region of a gene. They have predetermined tools that you can use on their website that makes it very user-friendly, so I can just plug in a gene sequence and it will give me 10 different primer sequences that I can select with confidence that it's going to help me achieve the results that I'm looking for in my assays.
The folks at IDT listen, and by listening, they can begin to understand what the challenges I'm facing are, and then say, well, maybe we could circumvent that problem with this approach, or maybe we could go over that problem with this approach. And what that does is open up opportunities so we can get to where we want to go more quickly, more efficiently, and with better success. In research, you do need to be creative, but the question is, where's that creativity come from? It comes from the environment. I have people around me, I have colleagues, the students, they ask questions. And importantly, students don't know what they can't do. And that is incredibly important for doing things that have never been done before. Lots of times in a lab meeting, somebody will be presenting, and they present their data and it looks really good. And then I'll ask, okay, let's all think about what experiment we could do that would kill this idea. If we can't kill it, it's got legs.
I get great satisfaction out of seeing people that I've helped train be successful. That's one of the most rewarding features of my job. We need people to get excited about it, to carry the torch and keep working in these areas, because I think our opportunities to do more and more for people with genetic diseases is just becoming greater and greater.
We get many rewards from the work we do. One is just the pure exhilaration of discovery, but also when you can combine that with what you've done for somebody, change their lives, that's incredibly powerful. So when patients, people with CF come here, tell us their stories, that leaves a lasting impression on everybody.
The mission of the Cystic Fibrosis Foundation is that we continue to do research until CF stands for Cure Found. And we all share that mission, and we all work every day to advance so that every single person with CF has a treatment option.